I am Dr. C.Swaminathan, Assistant Professor in the Department of Microbiology, St.Joseph College of Arts and Science, Cuddalore. Today we are going to discuss about nucleic acids. Nucleic acids serve as the repositories and transmitters of genetic information. Johann Friedrich Miescher in the year 1869 isolated the nuclein from nucleic acid of WBC. Nuclein is the nucleic acid. Nucleic acids are of two types, that is DNA and RNA. DNA is the deoxyribonucleic acid, RNA is the ribonucleic acid. Next we are going to discuss about the components of nucleic acids. Nucleic acids are the polymer of nucleotides, that is the polynucleotides. They are held together by 3' and 5' phosphate bridges. Nitrogenous base sugar and phosphate is called nucleotide. Nitrogenous base and sugar is the nucleoside. Nucleotide is thus a nucleoside phosphate. Now let us see what is nitrogenous bases. Nitrogenous bases are the aromatic heterocyclic compounds. Purines and pyrimidines are nitrogenous bases. Let us see what is purines. Purines are adenine and guanine. Pyrimidines are thymine, cytosine and uracil. DNA contains adenine, guanine, thymine and cytosine, whereas RNA contains adenine, guanine, uracil and cytosine. Purines are numbered in the anti-clockwise direction, whereas pyrimidines are numbered in the clockwise direction. The sugar which is present in the nucleic acid is the pentose sugar. Pentose sugar is the 5-carbon sugar. DNA contains deoxyribose sugar. RNA contains the ribose sugar. So this table shows the nomenclature of nucleotides. Next we are going to discuss about the structure of the DNA. DNA is a polymer of deoxyribonucleotides. James Watson and Francis Crick in the year 1953 proposed the double helical model of DNA, that is commonly known as the Watson and Crick's double helical model of DNA. James Watson, Francis Crick and Maurice Wilkins received the Nobel Prize in Chemistry in the year 1962. Structure of the DNA is comparable to the twisted ladder. In this picture you can see both the ladder and the DNA. Next we are going to discuss about the salient features of Watson and Crick model of DNA. So this is known as the bDNA molecule. DNA is a right-handed double helix. DNA consists of two polydeoxyribonucleotide chains. There are two strands which are twisted around each other on a common axis. The two strands that is present in the DNA are anti-parallel. That is, one strand runs in the 5' 3' direction and the other strand runs in 3', 5' direction. Width of the DNA is 2 nanometer. Each turn of the helix is 3.4 nanometer. There are 10 pairs of nucleotides in each turn of the helix. Each pair is placed at a distance of about nearly 0.34 nanometer. The two polynucleotide chains are not identical. They will be complementary to each other. It is because of the base pairing. Two strands are held together by means of hydrogen bonds formed by complementary base pairs. AT pair will have 2 hydrogen bonds. GC pair will have 3 hydrogen bonds. GC pair is 50% stronger than the AT pair. Hydrogen bonds are formed between a purine and a pyrimidine only. So the possible The possibilities are hydrogen bonds can be formed between adenine and thymine, thymine and adenine, guanine and cytosine and guanine. Complementary base pairing in DNA molecule proves the Chargaff's rule. The content of adenine is equal to content of thymine. Content of guanine is equal to content of cytosine. This is called a Chargaff's rule. Genetic information resides on one of the two strands of the DNA. So that is called the template strand or the sense strand. The opposite strand is called the antisense strand. There are two grooves in the DNA molecule. That is a major groove and a minor groove. Major groove will be wide. Minor groove will be narrow. Proteins interact with the DNA molecule at these grooves without disrupting the base pairs and the double helix. Next, we are going to discuss about the different forms of the DNA molecule. Helix type bDNA and aDNA are right-handed. ZDNA is left-handed. Overall shape bDNA is long and narrow, aDNA is short and wide, ZDNA is elongated and narrow. Diameter of the helix is bDNA 2 nanometer, aDNA 2.3 nanometer, ZDNA 1.8 nanometer. Distance per each complete turn bDNA 3.4 nanometer, aDNA 3.2 nanometer, ZDNA 4.5 nanometer. Distance between adjacent bases bDNA 0.34 nanometer, aDNA 0.29 nanometer and ZDNA 0.37 nanometer. Number of base pairs per complete turn bDNA 10, aDNA 11, ZDNA 12. Base pair tilt bDNA plus 19 degree, aDNA minus 1.2 degree, ZDNA minus 9 degree. Occurrence bDNA occurs in the hydrated condition, aDNA occurs in the dehydrated condition, ZDNA occurs in the dehydrated high salt concentration. Thank you very much for watching this video.